Alrighty, so hi everybody. I am Hailey Carter and I'm a master's student in the Hailey lab. Today I'm going to be sharing some preliminary results from an anatoxin exploration. I did this study originally as an undergrad on Lower Mill Pond in Brewster, Mass, and it was in 2019. I had originally focused on a couple different toxins, but just recently we decided to rerun all of the samples to see if we could detect any anatoxin, and we found some pretty interesting results. So my study mainly focused on looking at the aerosolization of cyanobacteria and monitoring that aerosolization through toxin levels. And aerosols were my main focus because they are an important and relatively unexplored route of exposure to a variety of different cyanotoxins. One of the cyanotoxins that I had originally focused on was microcystin, or MC, which is a liver toxin. However, today I'm going to focus on anatoxin, which is a neurotoxin that's also produced by cyanobacteria. Exposure to anatoxin has been reported as the cause of death for many different animals. So what commonly happens is the animal is exposed, and then depending on the concentration, if it's lethal enough, within just minutes, the anatoxin can affect the nervous system and cause respiratory arrest, which is how it's got its nickname of the very fast death factor. It's also common to see gastrointestinal symptoms with exposure. But both humans and animals are exposed to not just anatoxin, but other cyanotoxins through direct consumption, such as drinking water, eating fish that had previously consumed the toxin, and inhalation of aerosolized toxin. Cyanotoxins are aerosolized in two forms, the particulate and the dissolved. The particulate are the cyanobacterial cells in the air that contain the toxins within them, and the dissolved are free toxins that have dissolved in the water and then aerosolized. So to collect these aerosols, I use the CLAM, or the Compact Lake Aerosol Monitor. This is the maybe 5th or 6th revision of the collector that Dr. Hailey talked about briefly. So it uses a pump to suck the air off the surface of the water here, pulls it through a filter which collects that particulate, and then it bubbles the air through a series of 3 liquid traps. Because the anatoxin and the microcystin are both water soluble, the dissolved toxins are then resuspended into these liquid traps, and then I collect the traps and the filters and then concentrate them and test that for toxins using an ELISA. So anatoxin is produced by a variety of different cyanobacteria. You can see them listed over here on the right. In terms of degradation, anatoxin undergoes rapid photochemical degradation. Its half-life has been recorded anywhere from 1 to 2 hours, all the way up to several weeks. So anatoxin's rate of degradation is heavily influenced by pH and by light exposure. So those studies that had measured its half-life closer to a couple hours tended to use more alkaline conditions, usually with a pH around 8 or 9, and with more light exposure or a higher intensity. And then those studies that had recorded its half-life on the other side of the scale, around several weeks, had used conditions with little or no light exposure and more neutral or acidic pH. Of course, all of that data is based only on anatoxin that is in the dissolved form and it's in the water. So it's probably not very accurate to apply that degradation rate data to any anatoxin in the particulate form or in any aerosols. And that's just due to more exposure to UV rays without the protection from the turbidity of the water. So the degradation rate in aerosols really needs to be researched, and it's important because the longer anatoxin exists as a molecule in the air, the further it can travel and the more likely it is that someone is going to inhale it. Additionally, there is some evidence that aerosolization may increase at night. So if anatoxin aerosolizes at night and there's no UV rays to degrade it, that anatoxin will likely travel a bit further than if it had aerosolized during the day. In terms of guidelines, there are no estimated guidelines of anatoxin concentration for aerosols, but there are some varying recommended recreational and drinking water guidelines. Depending on the source, I found recommended limits anywhere from 1 microgram per liter all the way up to 80. And these aren't all the different guidelines that are out there. I just pulled a small sample to exemplify the huge range of recommended limits. So I had 3 main research questions going into this reanalyzing of my data. My first was to just determine if anatoxin is present in detectable levels in both the air and the water at Lower Mill Pond. I also wanted to explore to see if there was any relationships or correlation between anatoxin and microcystin, specifically in the aerosols. And then lastly, just as a very general exploration of anatoxin aerosols, I wanted to identify the next research that really needs to be done to help fill those gaps of knowledge on anatoxin aerosolization. For that first research question of detecting anatoxin in the air and in the water, I found that yes, we were able to detect it in both. This graph is showing bars of total aerosolized anatoxin on each date that I tested. And you can see that each bar is split up with the green portions representing the dissolved toxin, or what was measured from the liquid traps. And then that gray is representing the particulate, which was measured on the filters. Some literature suggests that anatoxin may exist more regularly within the cells or in that particulate form. However, I found that on average for my data, only 12.5% of the total aerosolized toxin was found in the particulate form, and that remaining 87.5% was in the dissolved form. This could be because maybe anatoxin exists more regularly in the dissolved form, despite what that literature suggests, or maybe the dissolved form just aerosolizes easier, so it exists more regularly in aerosols in the dissolved form. One other thing that I wanted to point out about this graph is that the aerosolized dissolved anatoxin spikes on July 5th, whereas the aerosolized particulate spiked on August 16th. And on that August 16th date, the particulate accounted for up to 48% of the total aerosolized toxin. I found this very interesting, just because it suggests that maybe there's different factors that are regulating production of dissolved versus particulate aerosols, which was something that I hadn't really considered before. And in terms of toxin concentration within the water, I didn't include any of those results just because I had very limited data points. I haven't had the chance to run all my water samples yet, and some of the ones that I did run were a little bit too concentrated, so they were above the detectable limit. So for now, the only point that I've concluded from that data is that there is anatoxin present in the water, but I don't have any estimates. All right, and here are my results from that second research question, which was, is there a relationship between aerosolized anatoxin and aerosolized microcystin? So I ran a regression to examine the relationship between the aerosolized microcystin on the y-axis and the aerosolized anatoxin, or on the x-axis, and the aerosolized anatoxin on the y-axis. I found that these two variables are in fact related somehow. I wouldn't say that there's a significant cause and effect relationship here, but I found this very exciting because it shows that anatoxin and microcystin aerosols are being produced at the same time, which suggests that the aerosolization of anatoxin and microcystin may be regulated by similar factors. And so just my conclusions from this preliminary exploration. I've concluded that anatoxin exists in detectable levels in the air and water at Lower Mill Pond, and the production of anatoxin aerosols coincides with the production of microcystin aerosols. And then for that third research objective, which was to determine some areas of anatoxin production and aerosolization that need to be studied in the future. There's countless topics and questions that I can think of that need to be explored, but personally, I think that the research regarding degradation of anatoxin in the particulate form and in aerosols is really important for estimating like risk of inhalation. And then also related to that, more research needs to be done to determine a more universal guideline for recreation, drinking water, and inhalation. And then up next for me, I would like to run another Anatoxin ELISA for the remainder of those water samples and some diluted samples from those values that were above the detectable limit, just because that will allow me to look for any relationships between the toxin concentration in the air and in the water. And then lastly, this upcoming summer, I am going to further explore these relationships and collect some additional types of samples. My hope is to develop a model to help estimate the production of both microcystin and anatoxin aerosols. And I'm going to base that model off of different environmental factors such as light, temperature, wind, maybe nutrient levels, also based on species composition, which I'm still devising my plan on how I'm going to do that, but I'm hoping to use some genome sequencing to identify the specific cyanobacteria species that are in both the water and the air on each date. And then lastly, some growth changes using pigments. I think that's all I have. So I have a quick thank you to everybody who was involved and some questions.